So thank you very much, Willie, for bringing us together, and of course, NBMA for hosting us. It's something that we've looked forward for a long time, and I do believe that at the end of the 3 days, we'll all go home better prepared to communicate biosafety. Now, I would like to just bring it closer home to those of us who are meeting this for the first time, and of course, try to bridge the gap so that we can move together. Now, when you talk about the international protocols that basically guide the biosafety matters, you will definitely be talking about the Cartagena Protocol on Biosafety. And the Cartagena Protocol on Biosafety is anchored on the Convention on Biological Diversity. Convention for Biological Diversity is also an international convention, an environmental convention, and it is the mother convention that brought forth the Cartagena Protocol. Now, it is very important to remind us that the Convention on Biological Diversity had 3 main objectives: the issue about conservation of biodiversity, the issue about sustainable use of biodiversity, and fair and equitable sharing of the benefits that accrue from biotechnology or biodiversity. And the realization of biotechnology as an important contributor to development agenda led to biotechnology being mentioned in the protocol. And of course, the protocol actually started the process for the development of the biosafety protocol. So biosafety protocol emanates from the convention, and the protocol, as it were, was adopted in 2000. It entered in force in 2003. And as we are going to have a discussion, you'll see how many of the African countries became parties to the Cartagena Protocol. And by becoming parties, they were then at liberty to develop, domesticate the protocol so that they are able to guide the development of biotechnology in their countries. So again, the CBD is the mother convention. It led to the biosafety protocol, or the Cartagena Protocol on Biosafety, and that is the international protocol that actually guides biosafety. So I'd like to spend a few times in defining what biosafety is. Now, biosafety is basically avoidance of risk to human health and safety, and the conservation of environment as a result of the use of genetically modified organisms. This is the definition that is provided for in the Cartagena Protocol. Basically, so the Cartagena Protocol is about safety. It's about safeguarding safety to human and animal health, and also safety to the environment. So whatever else that is provided for in the Cartagena Protocol, remember that it's about safety, and the objective of the Cartagena Protocol is an objective that Madam Agnes also alluded to, is to ensure there is adequate level of protection in the field of safe transfer, handling, and use of LMOs from the modern biotechnology, which may result into adverse effects on the conservation and sustainable use of biosafety or biodiversity, while taking into account risks to human health, human and animal health, especially during the transboundary movement. So the Cartagena Protocol is about safety. It's about safety in the transfer, handling, and use of LMOs during its entire phase of development. And we are going to discuss this even more as we dwelt into the more elements of the Cartagena Protocol. So again, the scope of the protocol is provided in Article 4. It's about application in the transboundary movement during the transfer, handling, and use of LMOs, and of course in the process ensuring that there is conservation and sustainable use of biological diversity while taking into account human and animal health. I think that's always discussed together, although in the protocol the focus is human health. Now, the protocol also defines living modified organisms, and for the sake of the mixed audience that we have, living modified organisms means a living organism that possesses a novel combination of genetic material that is obtained through modern biotechnology. So a novel combination, that is intentional combination of genetic material, and of course using modern biotechnology, and the protocol also defines what modern biotechnology is all about. And I would like to spend a few minutes just trying to highlight what modern biotechnology is about. And in a nutshell, the broader term for biotechnology, of course, is the use of biological systems for our own use, for human benefit. Any biological systems that can be used to generate a product that could have a commercial value or have some benefit to the human race, that can be the broader form of biotechnology. And biotechnology is with us in a number of forms, and many of us have applied biotechnology in one sense or another, or utilized biotechnology products. Now, when it comes to the modern technology that brings about the development of LMOs or GMOs, I'll use them interconvertibly, is where there is a transfer of genetic material from one organism to another to improve or to generate a modified product that has a desired trait. In very simple terms, you have a desired trait, you want to incorporate it into another organism, you can get that gene, transfer it into your target organism, and you will develop a desired trait. And that desired trait now defines a new organism, and that is the LMO that has been generated using genetic engineering materials. And the discussion on this can go on and on, but basically it is improvement of a crop, improvement of an animal using a genetic material for a desired purpose. And if you think about it, we have a whole range of possibilities, and I'm glad that Madam Agnes came before me. When this technology was being discussed, it had a great promise. There was a promise that we could develop crops that would resist diseases, crops that would resist insect infestation, crops that would resist drought, crops that would increase nutritional quality, crops. I mean, basically, a number of improvements could be made based on this technology, because it would involve just transferring the genes from one desired trait and introducing it into your crop or plant, and then you will be able to generate a GMO of interest. And I'll not try to use this, but basically you must have a desired gene. I've used an insulin gene. Insulin is a drug that is used in diabetic cases. If you identify the gene of interest, you identify a suitable, in science we call it a vehicle. You introduce that gene into the vehicle, then you have a recombinant which has your gene of interest and also your vehicle. And if you look at the next slide, basically once you have introduced this construct into a bacteria, you can then have your host cells with the bacteria multiplying that gene of interest, which you can then regrow, and you will be able to produce more and more of insulin as you desire. Now, in a similar scenario, in the case of insect-resistant crops, the focus is transferring a gene from a bacteria into a crop, and then you introduce insect resistant into the bacteria, into the plant, and then you use tissue culture to regrow the plant, and the plant will then have your BT in its genetic composition, and the plant will essentially have that product to resist insects. The same technology, but the applications can be so varied, and in the agricultural field, which many of our biotechnology, biosafety agencies were focusing on, we've had a whole range of products. For maize, we have had insect resistance, herbicide tolerance. Soybean, we have had insect resistance, herbicide tolerance. Cotton, insect resistance. Canola is an oil crop. Even the composition has been changed. Potato has been developed for virus resistance. Tomato, insect resistant, papaya, and banana. And this is a list that continues to grow. But in the African context, we have developed cotton that is insect resistance. That's what you've had and you will continue to hear. Maize, we have had maize that is resistant to insects and also drought tolerant. So there are more and more crops that are being improved with traits that have benefit to agricultural productivity and, of course, food security agenda. This is a development that continues, and it can never be exhaustive. When you look at the global arena, there has been several developments, but you would appreciate that increasingly herbicide tolerance has taken, many of the products that have been developed have been for herbicide tolerance. Initially, we had insect tolerant crops, but now we're having stacked events that has herbicide and insect resistance. So initially, we had single traits, okay, like insect resistance, including the BT cotton. That's a single trait. No, insect resistance is the blue one. Then the red one is a stacked event that has insect resistant herbicides. And of course, if you look at the trends, more and more of the stacked events are now what is in the market. And Madam Agnes highlighted how you have a stacked event in the maize, which has insect resistance and drought resistance. So increasingly, not single events, but more and more events are being inserted to have combined effect to basically have LMOs that have more than one gene of target. Now, in the global arena, of course, we have the leading countries, US, Brazil, Argentina, and Canada. We have developing countries, but it's always important that down here we have African countries, including South Africa is already here. We have Sudan. Now we have, of course, Nigeria. We have also Kenya, and we also have Eswatini among the countries that have also at least approved one commercialized product. And for most of the African countries, both Sudan, Kenya, it is BT cotton that has taken lead. Of course, for Nigeria, they have the BT cotton, they have the cowpea, and of course, the teller maize that have also moved to the next phase. So Africa is not left behind, but this is just to appreciate that the rest of the world have taken lead, and we are following. This is a report that was given in 2018 by ISA, and that was the last time they produced this report. And there has been, of course, several changes that you can peruse through just to appreciate. Now, talking about the biosafety protocol again, the Cartagena protocol, it has many provisions, and most of these provisions is basically what Madam Agnes alluded to in her presentation, including advanced informed agreements. If you are importing a GMO, risk assessment and risk management, information sharing, and the biosafety clearinghouse. This is an online portal where all parties are supposed to submit their approvals. There are provisions for handling transport packaging, capacity building, compliance, liability and redress, public awareness and participation, socioeconomic consideration, etc. I picked on a few of the articles just to explain what is provided for. Now, one of the very important provisions in the Cartagena Protocol is on Article 15. It is and Annex 3. This is about risk assessment, and it provides the basis for risk assessment, which has to be on a case-by-case basis. It has to be based on sound and transparent system, and it has to lack of scientific knowledge does not mean any specific consensus. And the fact that when you're carrying risk assessment, it has to be in the context of a comparison between the modified product and non-modified product, and that's what what we are calling the parental product. It has to be on a comparative basis. Now, the overall objective of risk assessment is to identify and evaluate the potential adverse effects of LMO on the conservation and sustainable use of biological biodiversity in the likely potential receiving environment while taking into account human health. The risk assessment methodology is also provided for in the Cartagena Protocol, which lays out how methodologically we go about doing risk assessment before you can conclude whether a GMO product is safe to the environment. Now, risk management is also provided for. If any risk is identified and it's possible to provide a management strategy, then a regulatory authority may decide that that risk is manageable and therefore it is not a reason to stop any particular approval. So risk management is very important. The Cartagena Protocol also provides for a socioeconomic consideration. In the Cartagena Protocol, socioeconomic consideration may be taken into account, but in most of our laws, we basically provide that socioeconomic assessment shall be taken into account, which makes it mandatory. Now, one of the very important provision of the Cartagena Protocol is public awareness and participation. Again, this has been alluded to very well, that in our decision-making process as regulatory agencies, we require the public to participate in the decision-making process. And for the public to have informed participation, there has to be public awareness. You have to go out of your way to explain what the technology is all about, and then that will enhance their participation in the decision-making process. Now, once you have signed the protocol, you become a party to the Cartagena Protocol, you are required to do certain things. One of them is to designate a national focal point. Agnes is a focal point for the Cartagena Protocol for Nigeria. And of course, you must also have a national competent authority designated, and essentially that allows you to set up administrative mechanism to regulate GMOs. So all the parties have an obligation that once you have signed the Cartagena Protocol, you must designate a national focal point, and you must domesticate the Cartagena Protocol. So domestication of the Cartagena Protocol means that you develop and enact the national laws that will guide the process. And I deliberately set out to use the Kenyan scenario to explain what this is all about. So that is the Cartagena Protocol. In the Kenyan system, there was a policy document that was developed back in 2006, and then we have a Biosafety Act that was enacted in 2009. Now, to implement the act, we had implementing regulations, the contained use, environmental release, export, import, and transit, and labeling regulations. Now, this was the Kenyan situation that domesticated the Cartagena Protocol to allow the functioning of the Cartagena Protocol. And the same applies to the Nigerian system. They would have the guidelines, they would have regulations to facilitate their functioning. And the intention of all those regulations is to cover the entire development phase of LMO. LMO starts from the laboratory phase, then you go through the greenhouse test, then you go through the field trials, and eventually you have environmental release. All these phases have to be captured in the development of the regulations or guidelines. And of course, the national performance trials, this normally is done by a different regulation. But in most of our countries where we have domesticated the act, the Cartagena Protocol, we have regulations, we have guidelines to facilitate our operations so that we can move from the lab and finally to the environmental release, and of course, the commercialization and cultivation, the whole process. And you're going to listen to some of the developers to explain to you that it's not something that happens in 1 year. It could take 10 years or more, but each phase has its challenges, and it is regulated through the various regulations that you have seen explained and also have alluded to. Now, the technology had its promise and its potential, but at the same time, it brought out a number of issues that, of course, were concerns, and it is not exhaustive. So one of the concerns was related to health, food and feed safety. Another issue was the impact on the environment, and lastly, socioeconomic issues. Now, all these issues have to be addressed before a GMO product is declared safe for environmental release. And I would like to just spend a few minutes to explain, for example, what happens in the Kenyan system and also in the Nigerian system, that before environmental release, food feed safety assessment is carried out, environmental risk assessment is carried out, and then there is socioeconomic considerations. In the Kenyan system, we have regulatory agencies that also make submissions, and also the public. So all this have to be taken into account in the decision-making process. In the Nigerian system, they have the technical committee. At the National Biosafety Authority in Kenya, we basically have expert groups that review applications. And then finally, the National Biosafety Authority, through its committee, decides whether an application is safe with respect to each of these parameters. So for purposes of environmental release in Kenya, the public participation is required to take effect once you have submitted an application. It is then published in popular newspapers, at least 3, and then the public is requested to make submissions within 30 days. We also hold a public forum, and then the comments that are received from that public participation has to be taken into account adequately before a decision is made on the safety of the product. But the focus is food and feed safety, environmental safety, and also the socioeconomic considerations. Now, this looks complex, but basically food and feed safety is focused on a comparison between the parent crop and also the gene from where you are getting it from, and then finally a comparison between the new crop and, of course, the parental crop. Now, and for food feed safety, the focus is on whether there's any toxicity or allergenicity. Are you introducing new allergens? Are you introducing new toxins in the new crop? And they are very well structured process of analyzing this to finally come with a conclusion that your product of interest is safe. And in doing so, you would also be interested in looking at composition analysis. Okay? Composition analysis is the major nutritional components, have you changed any, if anything? And the idea is the final product should be substantially equivalent to your parental crop. If it is maize hybrid A, you have modified it for insect resistance. The nutritional components should not change. The only thing that should change is the introduced gene, and you will then look at whether the introduced gene has any toxic effect inherent or any allergenistic effect, and this must turn out to be no before any product is released into the market. Now, the last slide that I have on the board is to basically to illustrate that in the African continent, almost all African countries are parties to the Cartagena Protocol. Out of the 54 countries, 51 of them are members to the Cartagena Protocol. And this, of course, emphasizes the fact that all the African countries saw the need and the possibility of biotechnology making an impact in their development. The only exceptions is Sao Tome, Equatorial Guinea, and Southern Sudan. They don't have, they are not parties to the Cartagena Protocol. Now, the countries in gray, they are parties to the Cartagena Protocol, but they have not domesticated the Cartagena Protocol. Domestication means having your own law and your associated regulations. Now, the countries that we have in green are countries that have domesticated, and they have some CFTs going on, the confined field trials. And the countries in stars are countries that have at least commercialized, so that includes Nigeria, of course, Burkina Faso, Ghana, Sudan, Ethiopia, Kenya, Malawi, Eswatini, and South Africa. So those are countries that have commercialized at least one product. South Africa, of course, has multiple products. But if you have a chance, this is a slide from Abuja, NEPAD. You can actually be able to get to know which countries have actually commercialized. There are just 6 of them. The other countries have some CFTs going on. Others have some domestication. But this is the African map that shows that indeed Africa from the very beginning realized the importance of the Cartagena Protocol, the importance of regulating the technology. Some have gone ahead to domesticate, others have moved on to have CFTs, and others have commercialized and are already reaping the benefits of the technology. And you have the DG here who can explain to you even more how some of the products have been achieved. And you have also representatives from Kenya who can also explain to you some of the benefits that have accrued from the development of that technology. I think that's my first slide, and I'm grateful for the opportunity. I'm ready for any question that you might have now or later. Thank you very much.